Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear students, we are in continuation of the chapter that is the cell, the unit of life, and we are going to discuss today in detail about the brain of the cell we call that is a nucleus. Okay, why we call a nucleus as a brain of the cell? Because all the activities of the cell is controlled or regulated by the DNA which is present in the nucleus. Okay, so we call it as a brain of the cell. So if we talk about a nucleus in more detail, then nucleus is a one organelle, a cell organelle, and it was first described by Robert Brown as early as 1831. In 1831, Robert Brown described this nucleus, and this is the Robert Brown, and this is the nucleus. Now, when we see the nucleus, generally it will look like this, and it has a two membrane. Okay, it has a two membrane, that is outer and inner. And apart from this, the outer membrane is continuation with the endoplasmic reticulum, and there is some time you will see the some pore in the this particular membrane or nuclear envelope. So we will discuss about the structure in much more detail. But nucleus was stained by the basic dye. Nucleus gets stained by the basic dye because it has a DNA as a main component, and DNA is acidic in nature. And it was the name as chromatin, and the content of nucleus which is stained, that is a chromatin, and the chromatin term is given by the Fleming. So it is a Fleming. Now the interface nucleus, if we say, means a nucleus of a cell when it is in non-dividing stage. So if we see the nucleus of a non-dividing cell, it has a highly extended and elaborate nucleoprotein fiber, means it is made up of protein and nucleic acid, and it is called a chromatin. So chromatin is what? Just a DNA and protein which is in a free form. It is not condensed. It is in extended form. It is not formed a chromosome. It is just like a thread, like. Then we call it as a chromatin. So see, this is a chromatin where DNA is coiled over the histone protein. Okay, so we will study this in the 12th one chapter, that is molecular base of inheritance, that this is the DNA. When DNA condense over the one protein that is known as a histone protein, and then the nucleosome is formed. Nucleosome when condensed, then it will form a chromatin fiber, and chromatin fiber is found in case of the nucleus, and when it get condensed, it is going to form a chromosome. So condensed form of chromatin is nothing but a chromosome which is found within this nucleus. So the interface nucleus, if we talk, having a nuclear matrix and one or more spherical bodies called a nucleoli. Interface nucleus, or the we can say non-dividing cell nucleus, has a one matrix and some spherical bodies that is known as nucleoli. The singular is nucleolus, and nucleolus is involved in rRNA synthesis mainly. So this is a nucleoli. Nucleolus is present here like this. Okay. Now the nucleus is covered by a nuclear envelope, and it is made up of two membrane which cover the nucleus. And you will see the space between these two membranes of a nucleus, and that space is 10 to 15 nanometer in diameter, and that space between the outer and inner nuclear membrane, it is called a perinuclear space. So see, this is the outer membrane and this is the inner membrane. Okay, and here you will see that this is the outer membrane and this is the inner membrane, and the space between this is the perinuclear space. Okay, it is a perinuclear space. Now the term chromatin was coined by Fleming, that we have studied. If we talk about the perinuclear space, which is present between the two outer and inner nuclear membrane, it forms a barrier between the material present inside and the nucleus, inside the nucleus, and that of the cytoplasm. Whatever material present within the cytoplasm and with the nucleus, if we talk between these two, it will act as a barrier. The outer membrane of nucleus remain continuous with the rough endoplasmic reticulum. That also we have studied. That outer nuclear membrane will form an endoplasmic reticulum, and it has a ribosome over this, so we call it as a rough endoplasmic reticulum. So when we see the nuclear envelope, you will see that the space is there, and this is a perinuclear space between the outer and inner membrane. And the nuclear envelope consists of two parallel membranes with a space between that is not a perinuclear space. So the nuclear envelope is interrupted by a minute pores. Some minute pores are also found in the nuclear envelope, formed by the fusion of the two membranes. Wherever the outer and inner nuclear membrane fuse, at that point you will see the presence of this pore. So see, this is the pore which are found. This is the nucleus, and these are the nuclear pores are found like this. So nuclear pores are the passages through which movement of RNA and protein molecule take place in both directions. We remember nuclear pores are there within the nucleus. In surrounding to that, there is a cytoplasm. So inside the nucleus and outside the nucleus, if the movement of RNA and protein is taking place, that takes place through these pores. Normally, there is only one nucleus per cell. Normally, there is only one nucleus per cell. And the nuclear matrix or the nucleoplasm, if we talk, which is present within the nucleus, it contains nucleolus and the chromatin. It contains the nucleolus and the chromatin. The nucleoli are spherical structure present in the nucleoplasm. There is only one nucleus per cell. Okay. And if we talk about the nucleoli, they are spherical structure present within the nucleus in the nucleoplasm. So if we talk about the content of the nucleolus is the membraneless structure which is found within the nucleus, and it is mainly involved in ribosomal RNA synthesis or rRNA synthesis. So if somebody asks what is the function of nucleolus, then you should answer that it is involved in rRNA synthesis. So if we take an example, the cells like nucleus, they are known as anucleated cells. And if we talk about the erythrocyte of mammals, that is RBC of mammals, and sieve tube cells of the vascular plant, that is pteridophyte, gymnosperm, and angiosperm. Mainly, angiosperm have a sieve tube. So in that you will see the absence of RBC, absence of nucleus. So absence of nucleus is the characteristic feature of this erythrocyte or RBC and the sieve tube cells. So if somebody asks what is the nucleopore, nucleopore is a nuclear envelope is represented by a minute pores formed by the fusion of the two membrane. Okay, so this nucleus is found absent in case of the RBC and the sieve tube cells. So you can see these are the RBC, these are the mature sieve tube cells, and in this you will see that there is an absence of nucleus. So mature sieve tube cells also do not have a nucleus, and you will see that the companion cells with the dense cytoplasm, they are associated with the sieve tube elements. Okay, and about sieve tube we have already discussed in the anatomy. Now, larger and more numerous nuclei are present in the cells actively carrying out protein synthesis. See, why? Nucleoli is what? Nucleolus, plural for the nucleolus. See, protein synthesis is carried out by the ribosome. And ribosome are made up of rRNA. And rRNA are synthesized by a nucleolus. Because of this, the cells which are carrying out actively protein synthesis in these larger and more numerous nuclei are found. But when we talk about the different stages of cell division during which cells show structural chromosome in the place of nucleus. See, chromosomes are not formed from the chromatin till the in a non-dividing stage, but when it is dividing, when it is carried out mitosis or meiosis, during this you will find that the chromatin is condensed to form a chromosome. Okay, and this chromosome will look like this. Okay. So chromatin get condensed and form a chromosome. In non-dividing phase, it is in the thread-like chromatin form, and in the dividing phase, it is like a chromosome. Okay. So chromatin, if you talk, it contains, or it is made up of, it contains DNA, some RNA will find, basic protein histone is found, and some non-histone protein are also found. So chromatin consists of DNA, RNA, histone, and some non-histone protein. Okay. So this is a DNA. Then now DNA is there. DNA transcribe and form a mRNA, because DNA have a coding information for the mRNA for the protein. And after formation of mRNA, it carry out a translation to form a protein. So this process is transcription, which is carrying out formation of mRNA from the DNA, whereas in protein formation from the mRNA, it is called as translation. So histone protein, if we talk, histone protein are like this, and over which the DNA is coiled and it formed a chromatin. If we talk about DNA, we know that the DNA has a deoxyribose sugar, and if it is RNA, RNA has a ribose sugar, and these are the structural component of the deoxyribose and ribose sugar. Dear students, we remember the ribose sugar is a 5 carbon sugar, and see, this is a ribose sugar. Okay, it is a 5 carbon sugar. One has oxygen, and this is the first carbon, this is second, this is third, this is fourth, and this is fifth carbon. Okay, and this is oxygen. Like this, the structure is HOH group is there, but at the second carbon you will see that OH group is present. But from OH, if we remove the oxygen, then it become a 2-deoxyribose sugar. So here it is similar to the ribose sugar, but here the oxygen is removed. So it is a deoxyribose sugar. Deoxyribose sugar is found in DNA, and ribose sugar is found in RNA. Okay, so in DNA, sugar molecule is a deoxyribose, whereas in RNA, sugar molecule is ribose. Then, if we see the single human cell, and if we talk about the DNA is a 2-meter long thread, and it is distributed among the 46, that is the 23 pairs of chromosomes. So 46 chromosomes are found in 23 pairs of chromosomes. If we talk about every chromosome has a primary construction called a centromere. Okay, so this is the what particularly a centromere, and this is the two chromatid. See, this is one chromatid, and this is another chromatid. These two chromatid are joined by the centromere, and these two chromatid are considered as a sister chromatid. So the primary construction is nothing but a centromere, and this chromosome look like this. So this is the centromere, and the two chromatids are joined. So chromatids are formed by the condensation of chromatin. On the centromere, on the other hand, if we talk about the centromere, on the centromere you will see the disc-shaped structure which is present, which are called a kinetochore. Okay, so surrounding to the kinetochore, you will see the presence of this surrounding the centromere. See, here it is a centromere region, and on the disc-shaped structure found on the both sides, that is called as a kinetochore. Okay, so this is what about the particularly we can say the nucleus thing. So whatever the important information about the nucleus and its about the structure, we have already studied the things. Now we have to note down some of the important points. So if we talk, we have studied that Robert Brown discovered this nucleus, and we have also studied that it is a double membrane bound structure. Outer membrane is in continuous with the endoplasmic reticulum. Then the nucleoplasm is there, nucleoli is there. All these structures are found. Okay. So we can say that nucleus is absent is RBC and prokaryotic cell, and particularly we can say the cell tube element. But multinucleated condition, so multinucleated condition when arise due to fusion of many cells then it is known as syncytium. So it is formed multinucleated condition formed by the fusion of the cells. But if the multinucleated condition arise due to repeated nuclear division without cytoplasm division, then it is known as sinusoid. Okay. The shape and size of the nucleus vary with the type and function of the cell. It can be rounded, oval, or disc shape. The structure diagrammatically we have studied, and we know that When we talk about the structure of nucleus, nucleus has four parts. And if we talk about these four parts, the number one is nuclear membrane, or nucleolemma, or karyotheca. Okay, so this three, we can say nuclear membrane or nucleoplasma or nucleotheca are there. And this nuclear membrane forms envelope of nucleus. So first thing is this. Second component is nucleolus. Third is nucleoplasm, and four is chromatin. Okay, these things are there. About nuclear envelope or nuclear membrane, we have already discussed. In prokaryotic cell, nuclear envelope is absent. We have already talked about the perinuclear space. And in nuclear membrane, we remember that inner membrane is smooth, whereas if we talk about nuclear membrane, that is interrupted by the, whereas outer membrane is rough due to presence of ATS ribosome. Okay, now they have a nuclear pore and exchange occurs through this, that all things we have discussed. Next, if we talk about a nucleolus, nucleolus carry out rRNA synthesis. And this nucleolus is membraneless, that we know. Okay. If we talk about nucleolus, it is discovered by Fontana. Discovered by Fontana, and term nucleolus given by Bowman. Okay. Okay. Then, if we talk about the size of nucleolus, vary depending on activity of protein synthesis of cell. Then nucleolus, if we talk, is, we have discussed, it is mainly involved in RNA synthesis. If we talk about nucleoplasm, this nucleoplasm is also known as, nucleoplasm is a transparent semi-fluid granular substance known as nuclear sap or nucleoplasm or karyolymph. And this nucleoplasm mainly made of nucleic acid, protein, enzyme, lipid, minerals, all things are present. Then we have a chromatin. And if we talk about a chromatin, this chromatin is a hereditary part of nucleus. It is hereditary part of nucleus. And chromatin is remain as an uncondensed network of filament or fine threads. And during prophase, it condenses to form chromosomes. It condenses to form chromosomes. Now after this, if we talk about a chromatin, this chromatin is differentiated into two regions. Into two regions. Number one, that is heterochromatin, and number two is euchromatin. Suppose this is the chromosome. In chromosome, if suppose we have understood that the chromatin is there, so chromatin is here tightly coiled, but here it is loosely coiled. Okay, so this tightly coiled region is known as heterochromatin. And this heterochromatin, if we talk, it is a tightly packed DNA, or it is a tightly coiled DNA is found in this heterochromatin. And if it is tightly coiled, the enzyme or protein which are required for the transcription or the replication are not going to easily interact with this heterochromatin. And if they are not interact, they cannot form mRNA and protein. So it is genetically and transcriptionally inactive. It is not able to form a mRNA because it is tightly coiled and it is not particularly have a space for enzymes to interact. And it is get dark stain. It get dark stain during interphase. But if we talk about this is a euchromatin, which is a loosely coiled. So if we talk about this is interphase, if we called about a euchromatin, dear student, euchromatin, if we talk, this is loosely packed DNA or it has a loosely coiled DNA, and this euchromatin, which is loosely coiled DNA, is transcriptionally or genetically active. And if it is transcriptionally and genetically active, means it has the ability to mRNA. Why? Because it is a loosely coiled, so enzymes can interact easily. And we stain during the interphase, it get light stain during interphase because it is loosely coiled. Now, if we talk, then this is the what particularly thing. Now, after this all structure which are present in this, the chromatin is going to form a chromosome. And when we talk about chromosome, it is condensed form of chromatin. But this chromosome, which are a condensed form of chromatin, which has a centromere, which has a kinetochore, that all things we have discussed. But based on the position of centromere, the chromosome can be classified into the four types. Dear students, be remember based on what? Based on the position of the centromere, that is the primary trunk constriction. This chromosome are of four types. And the first type is metacentric, another one is submetacentric, then telocentric, and then acrocentric. These four types of chromosome are formed based on the position of the centromere. And the disc shaped structure on the centromere, you will see that is a presence that is known as kinetochore, that already we have discussed. So first chromosome, if we discuss, that is a metacentric chromosome, then in this the centromere is present at the middle position and it forms a two equal arm of the chromosome. So when centromere is located in the central position, two equal arms of the chromosome are formed. So this is known as, because of this, it is known as metacentric chromosome. And metacentric chromosome like this, because the particularly here, if we talk, the chromosome produces two equal arms, and this chromosome is appear V-shaped. This metacentric chromosome appear V-shaped during anaphase of mitosis. Okay. Then after this, another chromosome are known as submetacentric chromosome, and this submetacentric chromosome has a centromere near to one end of the chromosome, and if the centromere is near to end, it result in one shorter arm and one longer arm. And this chromosome are known as submetacentric chromosome. So you can see that they have, these are submetacentric chromosome, and this is a longer arm and this is a shorter arm. So when one is a shorter arm, one is a longer arm, they appear L-shaped during anaphase of mitosis. And after submetacentric, acrocentric are there. Acrocentric chromosome, the centromere is situated close to its end, and when centromere is located towards the close to its end, it forms one extremely short and one extremely long arm. So your case, you can see that this is the acrocentric chromosome, and necrocentric chromosome, we can say one is very short arm and another one is the very long arm are produced. So this chromosome appear J-shaped during anaphase of mitosis. Then the fourth type of chromosome is telocentric chromosome, and in telocentric chromosome, here the centromere is situated at one end of the chromosome, that is a terminal chromosome. Terminal centromere is found. So when terminal centromere is found, this is C, this is centromere, and this is a terminal centromere. Okay. So this appear I-shaped during anaphase. So this is the what a particularly telocentric chromosome. Now these are all the telocentric chromosome, but if the centromere is absent, then it is known as acentric chromosome, and if there are two centromere, then it is known as dicentric chromosome. So we remember acentric means centromere absent, and dicentric means presence of two centromere. Okay. Now, apart from this, if you see some of the chromosomes have a non-staining secondary constriction. Secondary constriction, primary constriction is centromere, but some chromosomes have secondary constriction as well as, and this is present at a constant region. So this gives a appearance of a small fragment called a satellite. Okay. So this secondary constriction, it is known as non-staining structure. Okay. So chromatin appear as a bead on a string structure. Okay. So here first we need to understand this